Okay, good morning, everyone. A lot of old friends here, and really appreciate you coming. Welcome to session number 58, the Symposium to honor the life and career of Dr. Philip K. Russell. Phil Russell, or General Russell, as those of us in the military would always call him, a former president of the ASTMH, was a longtime leader in tropical medicine, global public health, health security policy, and the development of focus initiatives and solutions intended to prevent and to treat infectious diseases, whether in the military, academia, government, biotech, or nonprofit sectors. He was a clinician scientist, an organizational leader in the military and beyond, a practical academician, and probably most impactful, a mentor, probably for a number of people in this room. In all his activities, Dr. Russell really embodied the U.S. Army's values of loyalty, duty, respect, selfless service, honor, integrity, and personal courage. Our goal today is to honor Dr. Russell's incredible career, to reflect on his impact, and hopefully to inspire others to similar types of selfless service in global public health. Today we'll have 5 presentations that will highlight key areas that were relevant to General Russell's career: malaria, epidemic and pandemic response, emerging infectious diseases, dengue, and the greater intersection of zoonoses and human health. My co-chair, Dr. Gina Rabinovich, will present some perspectives on malaria and malaria vaccine development. Mike Levine from University of Maryland, who really provided the impetus, and shall I say the activation energy for this commemoration, will speak to COVID-19 in Africa. He's pinch hitting for Dr. Samba So, who, owing to some travel-related complexities, was unfortunately unable to attend. Tom Monath will speak on aspects related to Ebola vaccine development, and I think we're all aware that's a topic that continues to be very relevant as we follow and try to address the current events in Uganda related to Ebola Sudan. Scott Halstead will present some perspectives related to dengue pathogenesis, vaccine development, and probably more. And these are areas where Phil really made an important impact throughout the entirety of his career. And finally, Jim Leduc from UTMB will discuss the intersection of global health, zoonotic disease, and the foundations of One Health. And depending on timing, we'll probably have an opportunity for some questions at the end of each presentation. We'll play it by ear. So thanks again for your attendance at this event, and I will turn it over to Gina.
Thank you, Kent and Mike, for the invitation to come and speak on what has become an introspection of what has been learned and what we still have to learn. It remains the action agenda for 2022. Some of you will be very disappointed that I am not using PowerPoint. But instead, what I'm going to do is try to tell some stories that related to Phil, actually at the tail end, after his military career, and draw some reflections for the future of things that I think he would challenge us with. I met Phil Russell the fall of 1990. I started counting how many years that was. Whoa. After his career in the US military, and 7 years after he had served as president at ASTMH. I was already at NIAID and joined an National Vaccine Program Office. Bruce Gallen is here, and he's been the latest director. But this was 3 months. This was the US government office focused on the national strategy for vaccines. This was 3 months after the launch of the Children's Vaccine Initiative, promulgated, I think, in part by Seth Berkley, Rockefeller, and a broad array of partners. And Bruce happened to be in the room at that point doing something, so he got to sign it. The Children's Vaccine Initiative was unusual. It was not about a single vaccine. It was not about malaria or HIV or neglected diseases. It was rather an aspiration for the ability to reach and protect children globally with an effective, streamlined regimen of vaccines covering a broader array of diseases. We wanted more. We knew science could serve. It was probably a bit naive in its desired technical parameters, a single dose of a multivalent vaccine to cover everything. I think that was okay, but it was a response to the great stall in the global program, which had failed to be able to introduce the new, albeit more expensive vaccines like Hib, pneumococcal, and rotavirus vaccine. This was already 1990. HIB had been licensed in the United States since 1982, and there was no path forward to getting that pneumococcal rotavirus vaccines into the global program. So we convened. We didn't have a lot of money. We had convening power to bring a global group of immunization vaccine experts to chart a path forward. And we argued, plotted, questioned, and challenged every aspect of how this could possibly happen with no money. In 1993, when Phil had become, this was already, the founding president of the Sabin Vaccine Institute, he drew Bruce and myself, sometimes Amy, I don't know if she's here, into the Sabin-sponsored meetings at Cold Spring Harbor, tackling topics that were really not within the remit only of the military. How do you prepare for pandemic flu? Was one of the first ones. Can we develop a global vaccine policy? How do we overcome the challenges of introducing and using vaccine, introducing being a heavy-duty item, which we learned as we went along, but actually then using and sustaining vaccine in developing countries. This phase frames the interest that Phil was taking beyond the lab, beyond the military pathogens, financing, delivery of vaccines, preparation for a global pandemic. And Bruce and I staffed several of these and learned a lot about the challenges of translation, at least the meeting version of that. That would help. I was afraid to do it because I didn't know if I would make your system through. All right, thank you. During that time, I remember distinctly the moment that Phil was doing a global analysis of the pneumococcal vaccine landscape. This is not something, he knew, of course, of pneumococcal vaccines, but he went to every manufacturer, every laboratory. And I recall him roaring back into NIAID because with the statement, by the best estimate at that time, a multivalent pneumococcal vaccine was going to cost $7 per serotype. So that puts it at about the price of what each dose costs in the US or Europe right now. But his question was, wouldn't it be terrible if the vaccine was produced and the children that needed it most could not afford it, which is exactly what we were walking into. The pneumococcal vaccine story itself is long and it's detailed, and I can't do it justice here. But the learnings as a community continue to be valid. First, technology, first you have to prove the concept, but technology has to be optimized. Industry can't afford to continue making the kinds of investments in improving manufacturing and sometimes tweaking things, even though the manufacturing group doesn't like it, to optimize the vaccine. And here it included advances in conjugate design, selection of linker protein. One of the major companies closed down their conjugate program for 2 years and streamlined their program. But also the manufacturing processes that allow for very large scale. Beyond that, we learned how very hard it was to finance, to introduce, and to scale a new vaccine, particularly for diseases of children that were a diagnostic challenge. Who can see your rotavirus? Who can hear and distinguish between a cold and a pneumonia and a malaria? And are often not recognized as serious by parents, either in the United States or in other countries. And not least when diseases are not understood or considered a threat, at-risk populations are not necessarily eager to receive them. So you have end-user failure, you have development of technologies and design and schedules that need to be advanced that are expensive to do, and in the middle, you have limited resources. We also learned that the groups that know how to do this, not surprisingly, was industry, because this is what they have to do for their for-profit vaccines. And so we as a global community had not introduced, we were stuck with the same 6 antigens that had been in the EPI program almost since onset. We had not introduced a new vaccine into global health. It took a lot of work to develop the public sector pathway on how to do this and how to finance the work to do this, which again is what industry does. The kinds of interventions that were developed, and I wish I knew it was Oren was here, I know he's at the meeting, but including the ADIPs, Advanced Development and Introduction Projects or programs. Pneumococcal vaccine was led by Oren Levine. There were development of new financing tools for development and a revitalized EPI, and of course the enormous resources of Gavi that came along with multi-donor funding. The long-term result is part of a success story with many, many parents. Gavi, I looked it up, I think it's 2019, so it should be pretty current, is quoting a dose of $3 for lower and middle income countries versus a dose of $45 for rich countries. And this is the 9 or the 13 valent, and is it better? You can get all those papers. Second, there has been an uptake at significant scale in the poorest countries, and this is driven by revitalized programs. And I'll comment on that at the end because COVID has had a huge impact. And a third is the recognition that the best science can serve health only when it is scaled, and that many partners, including countries and industry, are key members of that evolving social construct. So in the midst of this change, this requirement for advancing a single vaccine, let alone malaria, TB, HIV, one evening, Phil went to dinner. This was a really good thing. He often went to dinner, but this was a special dinner at the big house in Seattle, so we're back where it all began. He was not the only one there, and I've not seen detailed notes of that event. I bet you that exists someplace. But he came back, and he's telling me about dinner, and I'm complaining about NIH, and then he tells me, but there's dinner with Trey. And I ask, who's Trey? And it turns out Trey was the nickname the family used for Bill Gates III. That's the Bill that you know. And it marked the beginning, that dinner marked the beginning of a commitment by the Bill Melinda Gates Foundation to develop immunization with the funding first of the Children's Vaccine Program at PATH. I don't know if anyone remembers that, but merging into the creation of Gavi, and later a focus on developing vaccines for pediatric diseases and beyond, where inequity is unacceptable, including HIV, malaria, and TB. Thus was created around, following that, the initial support from the Foundation for IAVI, but the creation also of Aeras, MVI, and other product development partners. So let's talk about how this influenced malaria. See, I told you I was going to get to malaria.
I was worried.
No, you weren't. Because I told them I would, and because I continued to work in malaria, though less tightly linked to vaccines. Malaria at that time, so now we're talking, the big house was around 2000, right? The creation of the foundation. So it was all right about that time. And malaria at that time had a mortality of, they cited 1 million children per year. But who knew? It was modeled for half of the African countries. Maybe 240, 250 million cases, 1/3 of the global population endemic for the vector with people at risk. Little data from the highest burden countries. Existing tools were microscopy for diagnosis, chloroquine treatments, and integrated vector control were possible with insecticides. That was it. And the tool set that we have now was financed very early on that with rapid diagnostic tests, with long-lasting bed nets, not things that you had to dip, with effective treatments with artemisinin. Now, having a vaccine for malaria was an audacious target. It was the sort of thing where people would pat me on the head, and I actually took the job on as the first director. But they would pat me on the head and say, it's really nice that you're doing this, and that Bill's funding this, and you should get your funding from Bill. But they had reason to question this. The vaccine for a parasite had not been created. Everyone knew already then, without the tools that we have now, that the immunology was complex, the epidemiology was complex. And much of the effort at that time was research-based. Not that that is a bad thing. We have had great investigators generate candidates and product development pathways, whether it's been inside the military or outside. And that included NIAID, Lou Miller's lab, academia, Nussensweig, who patented and identified CS, a variety of donors, and then universities. Very few companies were involved. And again, it was two things. Never been done before, well, three things. Never been done before. It's going to cost a lot. Where's the money going to come from? And where's the science? It also was a perception at that time. In 2000, I went to a meeting in Australia. And I talked about the need to have the capacity to conduct GCP trials in Africa. And I was literally laughed out of the room. Oh, I didn't leave, but they laughed at me. Because this was conceived of as an overwhelming challenge. And GCP is not fun. GCP is not fun, and every developer in this room knows that there are reasons why it's not fun, and it's a useful tool. But it was also at a time when the first vaccine that had been tested out of Colombia in Africa had already failed. So there was a sort of a aura of doom around our capacity to actually engage this with good science. Following that dinner, Phil was able to make the case that the promise of emerging technologies and the intense need made this a worthwhile area for support. And at that point, Rip Ballou wrote a 10-page, double-spaced, because I've seen it, I probably have it somewhere, concept note that led to the award of the first $50 million for MVI at PATH. And with that money, we did the same thing you do at a party. You start having a party. You talk to everyone that could have a construct, an antigen, an adjuvant. You visit them, you see the labs, you see the facilities, and could potentially partner with their vaccine. We documented that half of the global effort at that time was being spent, and I'm talking about energy, labs, constructs, any way you measure it, on a single blood stage antigen called MSP1 that has subsequently failed in trials. And with an incredibly complex patent map. Someone had told academia that if you didn't patent it, you couldn't get industry to manufacture it and to sell it. So everyone with overlapping patents, we showed that patent map to the patent people at Rockefeller, and they almost fell out of their chairs laughing. There were very few companies engaged, and even when you went and say your adjuvant or your technology could be applied to this, sometimes they were interested, but quite often not. They had their own boards and their own mandate on things that they needed to be doing. But MVI in the first 3 years, generated and reviewed and funded a portfolio of vaccine investments, and by year 5 had 2 pediatric trials being implemented. One was MSP1, which failed, and which was found to be negative. It didn't fail, the trial was well done. And the other one was RTSS that met the minimum criteria for moving forward. Now the same questions remained. At every single negotiation, which was not a fun thing to do, but I got better at it, and even more important for this kind of vaccine compared to pneumococcus, because there was no upside of a global market. There was just a southern market. About scale and price and access and kind of commitments that people could make, particularly with something that had never been in humans. And it was a really tough point of negotiations. We struggled with it, and I don't think we have the right recipe yet. Phil, at this time, was advisory on the advisory group for Malaria Vaccine Initiative, but he was also on the first boards for IAVI and Aeras, so really deeply involved in the scope of vaccine development across the three hardest, and probably other things. I don't know everything that Phil was doing at that time, as a capacity to engage was enormous. RTSS has continued through phase 3 trials, documenting partial efficacy, to a pilot implementation and evaluation in 3 countries, which was presented or will be presented, I think, yesterday, and with recent adoption, I think last week, of as national policy in Malawi. After that, and hot on the feet of that success, on the progress, on something, is the R21 vaccine from the Jenner Institute, irradiated sporozoite-based vaccines, and more recently, not even in a phase 1 trial yet that I know of, is the potential for evaluation in the rapid mRNA platform as a commitment from one company that has made enough money from COVID. So these are really good things. It's a partially effective vaccine, and right now we have severe, where I didn't want to be 22 years ago, funding a concept in which we wouldn't have the scale of production, question about affordability. Those as two key elements of it's great to have great science, but if you can't push it forward, you can't make it available. And it's been a rough path, and I've tracked that path. And there are many guilty people in that pathway in terms of how things got slowed down, not least which was a pandemic at the tail end. But the key problem is really the lesson here. It's not a broken business model. It's one that never existed. It never had to exist. The old vaccines came out of laboratories, were produced by someone, they were good enough, they worked really well, they had very high efficacy. We went forward, at least 100 years later, it looks easy. But it's a missing business model for product development. It ranges from the minor, the lack of comfort we have sometimes with having companies in the room, even though we need them as part of the effort to have quality scaled product. But it's also what's the right recipe for what industry should be putting in, what the country should be putting in, what donors, philanthropy, research institutions should be putting in. And with the recognition that development is extremely expensive, especially if it's GCP, large clinical trials. And there's been lessons learned. Steve Hoffman has amazing partnerships with Africa in which the countries are funding a great number of those trials. And there's been a lot of success in engaging manufacturers. Serum Institute of India is a key example with Meninge, and the latest is announcement for pneumo. They are targeting a $2 pneumococcal vaccine. So the potential for progress is critical because malaria as a field has met the capacity, has met and been challenged by, this is what I mean, the capacity to continue to impact due to resistance, due to partial effectiveness of all form of vector control, due to delivery challenges. Right now, what Gavi is saying is that 37 million children are in the 0 vaccine group. It's not that they're delayed, it's that they've made a clinic visit. They have 0 vaccines. And in discussions with UNICEF, it's really clear that those children that have 0 vaccines, that's not an accident. It's not a mother or a family that said, we don't want our child to have vaccines. That's a global phenomenon for another topic. It is children that are not linked to the healthcare system, who are not receiving BETNAs, don't have access to treatment, and are probably not getting adequate nutrition. So back to malaria, the potential speed of progress is critical. We've met that capacity, but we will miss the 2030 impact goals. Now, Phil's role in all of this was imagination, creating and working on concepts that had not existed. Just because something had never been done, he didn't hesitate to jump in to figure out what needed to be done next. Fearless tenacity. I think looking back, he wouldn't have called it that. In fact, he would have resisted us calling it that. But looking back, his angst at not being able to scale or deliver a pneumococcal vaccine when the science was right really were concerns with equity. How can our children survive, receive vaccines, receive care, knowing that the rest of the world does not? And his focus on children. Who knew? I mean, he has his children, beautiful family, but I'm talking about as a target for product development. And that shows his flexibility and understanding not what he can do with what he knows, but how he can apply what he knows to tackle new problems. And his ability to work with a quite diverse group of partners, including industry, WHO, governments, NGOs, and all on a robust scientific base. We were fortunate, the community, Bruce, Teresa Guado, Amy Batson, many others, myself, not to be advised, because he always says, no, I'm not going to give you advice because you might do what I tell you, and then if it doesn't turn out, it'll be my fault. So he never gave advice, but there was a chair next to the grill, and he would be cooking the skirt steak that he would cook, and Connie would be inside, and a variety of people sat in that chair, talking through their woes, listening to him, not giving you advice, and then kind of knowing what you needed to do afterwards. It was pretty amazing. And I was one of those. I was fortunate to be one of those. We were able to participate and come in at the beginning of a huge global movement in which Phil was often the leadership voice, where he forced us to question assumptions, to raise our voices, and to commit to the long-term impact that can be achieved, improving the health of the most vulnerable. There is still much work to be done. There's only one thing he failed to teach me, and that's how to salute. But then I wasn't Army, so that's probably acceptable. I think Connie is listening to this, and thank you, Connie, for letting him spend so much time and effort on the global effort, and to Katie, Ellen, and Richard and their families because of his travel schedule and him being so committed and gone so much. But I have to tell, he was so proud of you and was not, he spoke about you often. Thank you, Phil, for teaching my mom that a knee replacement was a good idea by showing her you could do it by dancing a jig in front of her. She had the surgery. She's fine. We were so fortunate to have a bit of him in our lives, and now we have to forge onwards with a bit of his voice in our heads, not telling us what to do because he wouldn't do that, but challenging us to do the right thing and to bring the best science to the table. Thank you.
Thanks, Gina.
It should be working.
Well, oh, there it is. Okay, so I think what we'll do is, in the interest of time, if there are any questions, we'll save them to the end so we can get through all the presentations. So next up is Dr. Mike Levine from the University of Maryland, who's going to talk about COVID-19 in Africa.
Okay. Good morning, everyone, and thank you for coming. I'm filling in for Professor Samba Sow. Yes. Will you advance them, or do I advance
If
them?
you can do arrow keys to go forward. And then which one is your slides?
Number 4, Samba Sow. No, not that one.
Not that
No,
one. Oh, sorry.
one above it.
Okay. Just arrow key forward to go forward.
Okay. I am literally a last-minute fill-in for Professor Samba Sow, who, because of a personal conflict, will be a day late in arriving, and I will do my best. Samba Sow and the late Phil Russell share a number of characteristics. Both are born leaders. How do you define leadership? I would say by knowing Samba Sow, by having known Phil Russell, there are models. A few years ago, our society asked Samba Sow to give the commemorative lecture. And I introduced him at that time, and I will share a couple of comments from that introduction. First of all, Samba is a fellow of the American Society of Tropical Medicine and Hygiene. He's the Director General of the Center for Vaccine Development of Mali, a place where a number of individuals in this room work. He's the Regional Advisor for Africa on COVID-19 vaccines to the Director General of the World Health Organization. He's a past Minister of Health at a young age of Mali. He's a recipient of the Légion d'honneur of France and of the Ordre du Mali. He has been voted an international member of the National Academy of Medicine, USA. He's a professor of medicine at the University of Maryland School of Medicine and a wonderful teacher when he comes through. I'll tell a little bit about the young Samba Sow. He grew up in very humble environment, mostly in a place called Jikaroni Para, an area of Bamako. His mom worked at the local leprosy institute, the Institut Marchou, and his dad was a driver for the U.S. Embassy. Their salaries, though, had to support the extended clan. Samba went to school, grade school, that had no roof, it had no walls, it had no chairs, it had no desks. His desk, if you will, was a stone. It was his stone. It's where he sat every day. This young man went on to go to high school and graduate the first of his class, the beginning of a pattern. He went to the School of Medicine in Mali and was first in his class. He received a scholarship to go to the London School of Hygiene and Tropical Medicine to get a master's in epidemiology, and was first in his class there. And you'll notice where Samba is standing, right in the front and in the middle. Wherever he goes, whatever group he's in, Samba emerges as the leader. What are these qualities? But these are ones that he shared with Major General Philip K. Russell. We hired Samba when the University of Maryland was starting a field unit, a long-term field unit in Mali, and I asked the late Ogabara Dumbo if he could recommend someone, and he recommended this young man, Samba So, and 2 years later, he was able to get a fellowship to go to the advanced vaccinology course in France, and Annecy, France, and once again gets the award as one of the outstanding students at that. I'm going to speak a little bit about Ebola, hopefully without overlapping in any conflicting way with the next talk. But this was a dress rehearsal for COVID in many ways, for Mali, for the Center for Vaccine Development of Mali, and for WHO. This devastating epidemic of Ebola broke out, in particular in 3 countries, Guinea, Sierra Leone, and Liberia. As of March 11th, of 2015, more than 24,000 cases, 10,000 deaths, and a destruction of the infrastructure of healthcare delivery. Nurses, doctors, everybody who are at health centers taking care of these patients, a very high attack rate, a very high case fatality, and a generation of healthcare providers lost their lives in a place where there was a shortage of such individuals. This catastrophe was ongoing for a number of months, and a lesson was learned. It didn't become a public health emergency of international concern, a FAIC, until cases appeared in the USA and in Europe. Then it became a FAIC, and that kicked in the proviso that all nations, including high-income countries, must contribute to help control this problem. In that colored area in the southeast of this map, you see where the Ebola arose and then spread to all 3 countries. It's a site where those 3 affected countries meet. So come July 2014, suddenly there's a clamor for a vaccine to prevent this disease. And on the shelf, because of a civilian biodefense desire for an Ebola vaccine and a military defense desire for a vaccine, there are 2 vaccines sitting on a shelf that could be activated. One had been given to a single human being, one had been given to no human beings at this time. I will speak a very little bit about each, and again, I hope not to overlap. The World Health Organization had to somehow activate a means of rapid testing of these vaccines, and it did it in a very informal but very effective way by calling people in Africa and USA who worked to be able to see if they could drop other things and begin phase 1 and 2 testing of these vaccines. And 2 consortia were formed. One had the Center for Vaccine Development of Mali as its center, and that was the vaccine from the Vaccine Research Center, the chimp adenovirus 3-based vaccine. And that collaboration included Adrian Hill from Oxford University. It included folks at CVD in the University of Maryland, and folks at the Vaccine Research Center at NIH. And rapidly, CVD Mali cranked out a phase 1 dose escalation study and showed the safety and the immunogenicity of that vaccine. And by this time, because of interventions, including from the US military in Liberia and from UK groups in Sierra Leone, in those 2 countries, the transmission of Ebola was greatly diminishing. There were not enough cases in those countries to do an efficacy trial. But there still was in Guinea. Guinea was kind of the orphan. It's where the epidemic began, but while the other 2 countries were adopted by external sources, Guinea was not. And so Norway, Canada, and WHO went to fill that gap, and they tried to set up a phase 3 trial of efficacy of the vesicular stomatitis virus vectored vaccine. And the African Union made available health care workers who could participate in this trial. This was a trial that was based on the smallpox eradication concept of finding cases and then doing a ring vaccination of the contacts around the cases. And in this trial, the rings were created, and they were then randomly allocated to have immediate vaccination of the contacts who would be at risk of getting Ebola, or a 21-day delay until they got it. So ethically, everybody was getting vaccine. Some were just getting it sooner than others, and if you weren't in the trial, you weren't getting vaccine at all in these places where there was still Ebola. But these healthcare workers from the African Union had never participated in research. And so they had to be trained in good clinical practices, and they had to have that done in a couple of weeks. And again, CVD Mali, CVD in Baltimore, working closely with World Health Organization, Norway, and Canada, filled that gap. When the field trial started, there was 100% clinical monitoring. And you can guess there were many, 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 many mistakes. And those data were not going to reach regulatory approval at the beginning. The clinical monitors were magnificent. They were teachers as well as monitors. And these folks who had just learned the concepts, theoretically, of good clinical practices, GCP, they got better and better and better. And at the end, the monitoring showed that that trial team had as good a record as any experienced clinical trial team. Anyway, a couple of pictures here of what it was like in the field. In the field trial, Anna Maria Henao Restrepo from WHO spent months in the field. This was her baby, the Lancet publication of the high efficacy of doing ring vaccination around cases. At the same time, back in Mali, there was an outbreak of Ebola. And Mali and Guinea are, there's a border. The border is completely porous. There was every reason why Ebola should have exploded in Mali, but it didn't. It didn't because there was a leader. There was a special person, Samba Sow, who organized case identification, who did shoe leather epidemiology to find the contacts of all those cases, and then to quarantine every one of those cases and contacts, and to make sure that those people who were quarantined stayed quarantined and were fed. This was amazing. This went on for weeks. And Samba was a unique experience in Mali to stop an Ebola outbreak in its tracks. Towards the end, on the bottom right there, you see a picture of Margaret Chen, the previous Director General of Health, WHO, who went to Mali specifically to give an accolade, a shout out to Samba for what he did as an inspiration. Quickly, I'm going to go through a bit of COVID now. Think of Ebola and that outbreak and the interventions and what had to be done as a dress rehearsal for what happened with COVID. So when COVID hit a place like West Africa, Mali, one of the 6 or 7 poorest, least developed countries in the world, it hits in a place with a high burden of endemic infectious diseases, where the expanded program on immunization is stressed to deliver vaccines, where surveillance outside of population centers is very, very meager, and where access to health of any kind is a problem, particularly with nomadic populations. Early on, there were reports from Africa that Africa was being spared of COVID-19. And that turned out not to be true when you establish surveillance and when you finally get tests into places in Africa to look for COVID-19, and when you can do serology to look for antibodies. If you look on the left here, you see that by December 2020, Samba says, about 1/4 of the population had undergone SARS-CoV-2 infection. Africa was not being spared. Deaths and cases and inapparent infections were early on just not being detected. And because of the great infrastructure, the WHO's Solidarity Trial for Vaccines, Samba and his center were selected as one of the places for the Solidarity Trial. Because vaccine wasn't available at that time in Africa, new candidate vaccines, better suited in some ways for use in Africa, were put into placebo-controlled trial, and then were put into a trial where, like in Guinea, there's a delayed randomization. You get vaccine immediately, or you get vaccine in the trial after a couple of weeks. And in those studies, the incidence of confirmed cases becomes very clear. I don't see those colors, so I'm going to pass on that slide. So surveillance is a big problem. Surveillance to Samba, just like delivery of vaccines, this is like religion. It's, you have to do it right. You have to do it, you have to know what's going on. And there were many, many barriers to surveillance. But the greatest inequity, the one that bothers Samba and other health leaders in Africa, is shown in this slide. On the left is the coverage of folks who've gotten at least 1 dose of vaccine as of March 2022 in the high-income countries. And that percentage that didn't receive vaccine, in many countries, it's not because of supply. It's because of vaccine hesitancy. It's a different question. In the upper middle income, it's a lower percentage, but clearly higher than on the right. And the lower middle income goes even lower in terms of coverage. And then the low income countries, only a few percent as of March 2022 had gotten vaccine. And in Mali, it was really about 5% at that time. The consequences of this inequality is so many deaths that could have been averted. It's a widening of the economic gap that these countries face to begin with, and the possibility with such ongoing transmission that new emergence can appear. And Samba points out that in place like Mali, they need therapeutic intervention against early disease, because once it progresses, there's not the infrastructure of intensive care or even hospitals. There are very few hospitals in Mali. One has to nip and interrupt the progression of the disease early. And Samba likes to say that enabling health goes a long way. And there has to be plans for the next pandemic to get diagnostics, vaccines, and other materials such as personal protective wear to places early. Samba ends in his slides with what's next, and he believes that a commitment to public health is a commitment to optimism, and it's very, very much based on delivery at the local level. I have done my best to fill in for Samba, who is, you know, those of you who know him, he's a spellbinding orator. And anyway, thank you.
Thank you so much for stepping in. Thank you so much for stepping in, Mike. And on a slightly different approach, I hope, but the next speaker is Tom Monath, who is going to speak about progress and challenges and developments of Ebola vaccines.
Thanks, Gina. Yeah. Well, I know this is supposed to be a scientific symposium in Phil's honor, but I do have to provide a few personal reflections. I think I met Phil about 45 years ago at the first TropMed meeting that I ever attended. And since then, he was an integral part of my life and my career at every stage. I worked at CDC. He was an advisor. I came out every year for a review. And then I transitioned to the Army. I was, even though he had a long line of colonels darkening his doorstep every day, he always made time for me. And then he was an advisor to two companies that I worked for, and particularly the last one, a Canvas, where he, with D.A. Henderson, was in charge of trying to bring a smallpox vaccine through after 9/11. And we were working on that, and I used to go down to Washington nearly every week, and he was so interested not only in the policy but in solutions, and he wanted practical solutions for everything. He was very interested in vaccine technology and could talk at any level, and I owe him an amazing debt of gratitude as my mentor over that long time. So I'm going to talk a little bit about Ebola. I think Phil was always in the background here and played an important role in the development of countermeasures. And I'll go through a little bit of the history. There will be some overlap with what Mike Levine talked about, but that's all right. Principle of advertising, the more times you show something, the more people remember it. So the history here goes back to 1967, as I'm sure many of you know, when the Marburg virus, a related filovirus, caused an outbreak in Europe among laboratory workers and had been imported in monkeys from East Africa. And then about a decade later, Ebola was discovered in Zaire and in Sudan. And Carl Johnson, who still with us, unfortunately not here, shown in the picture, had a lot to do with the original investigations. Over the years, there were sporadic cases of this disease, relatively small outbreaks in sub-Saharan Africa. But there was investment made because of its obvious biodefense implications in research. And that included development of animal models of Ebola and research on vaccines, including much at USAMRIID, where Phil had oversight as commander. But there was no commercial interest. It's a disease of low incidence and in economically disadvantaged markets, but the research supported on this disease was critical ultimately to control of the outbreak when it occurred in West Africa. So the first vaccine publication was in 1980, so it's only a few years after discovery of the virus, and it was inactivated vaccine, didn't work very well, published in the Lancet in 1980. These are the outbreaks in that era from discovery through before the West African epidemic, and you could see that there were these scattered outbreaks only in Central Africa, really, where it was originally identified. Now, during that period, a number of vaccine candidates were investigated at different platforms. Inactivated viruses, recombinant VLPs, DNA, mRNA wasn't around then, and vectors of various kinds, adenovirus, and parainfluenza virus, vaccinia, VEE, replicons, and VSV. None of these reached the clinic in that era. There were a number of milestones, however, that were really important to the acceleration of events after the West African outbreak occurred. And they go back to the mid-1990s when Jack Rose at Yale developed a reverse genetic system for vesicular stomatitis virus and showed that it could express foreign glycoproteins. And then, I'm not going to read all these, but you see a number of important steps validating that platform in animal models and investigating adenovirus vectors as well, principally at NIH under Nancy Sullivan and her group. And a lot of the credit, however, on Ebola progress really goes to Public Health Canada, where they had started a commitment back in 2005 to develop a vaccine against Ebola and invested heavily and even had the foresight to make GMP manufactured vaccine in around the late 2000s. In 2008, this little company, Newlink, which I joined after the epidemic started, acquired a license to it from Public Health Canada, and they, at the time, there was just a burgeoning interest in government contracts for biodefense. And so that was their mission, that was their idea. They would get this technology and pursue some contracts from DOD, et cetera. So that's the history there. And then, of course, events unfolded in 2013-14. As you've heard from Mike Levine, I won't repeat this, this dramatic outbreak in a different part of Africa, 28,000 cases. There's interhuman spread, very mobile communities and densely populated parts of West Africa, and of course exported cases too. And this accelerated vaccine development, and fortunately, large companies became involved. So they saw that they needed to step up to the plate. There was still an uncertain market, but a very important opportunity for big pharmaceutical companies to play a role in containment of this disease. And Merck and Johnson & Johnson, GSK, became involved in the vaccine development effort. After the epidemic started, these are just some important dates, but you could see things rolling out rapidly month by month after the start of the outbreak. And Newlink got contracts from DOD and then BARDA to manufacture the VSV vector vaccine. And then in November of 2014, Merck sublicensed the technology. And with their tremendous scientific expertise and power, they were able to really push this vaccine forward. The first phase 3 trial started in February 2015, so it was really only a year after the outbreak got underway. And these were the leading vaccine candidates at the time. Merck, with the VSV vector, ultimately licensed as Ervebo in 2017. They began phase 1 trial in October of 2014, and phase 3 started in Guinea in 2015. So this unfolded very rapidly, and you can see the pace towards licensure. 2 of these vaccines ultimately approved by major regulatory authorities. What's really interesting, and the point that Mike Levine made about the role of Ebola in readiness for COVID is made on this slide. If you look at the blue line, this is the probability of a vaccine reaching market by the clinical phase. And you can see it's about 10 years. That was kind of the average for most vaccines. Ebola broke all records, 3 years to licensure. And then COVID came along and less than a year. But using 2 of the platforms that were used to develop Ebola vaccines, as you'll see in a minute. So this is the VSV platform. It's really interesting. It's a rhabdovirus. So those of you interested in arboviruses, this family is, contains many viruses transmitted by insects or rodents. And Jack Rose was able to, use this reverse genetic system to put in a gene, in this case, the Ebola glycoprotein, and it's displayed on the surface of the virus particle. So you now have this pseudotyped virus. It's still replicating, and it looks just like VSV under an electron microscope, but it has the protein of the desired vaccine on its surface. This is a slide from Merck, our colleagues at Merck, just showing the incredible partnership that evolved to facilitate clinical trials of this VSV vector vaccine. You can see trials in the United States and Europe, Africa, phase 1, 2, 3, and this is really a remarkable effort when you look back. Nothing really precedented in history before this period of time. There were 3 phase 3 efforts, NIH in Liberia, called the PREVAIL trial, and that investigated the VSV vector as well as the chimp-ad vector, looking at safety and efficacy and immunogenicity, starting in February 2015. The Guinea study that was successful, sponsored by and led by WHO, about 5,800 subjects. It started in March of 2015, and then the STRIVE trial, CDC led the way there in Sierra Leone, and that was a randomized trial of immediate versus deferred vaccination. And so the 2 of these vaccines, the Ebola vaccine was successfully licensed by FDA and EMA. The chimp-ad vaccine not, but the development program here was really instrumental in preparing for COVID. These are the epidemic curves in the 3 countries, and Mike Levine mentioned this also. The vaccine effort got underway, phase 3 effort on the downslope of the epidemic curve. That's always the way things happen. And that's really the impetus now to try to shorten the time between identification of an pandemic threat and introduction of a vaccine to, the target is 100 days, which would have, if successful, probably with the existing platforms, would have initiated pivotal trials at the peak rather than on the downslope. I think I'll skip this. This is a description of the cluster trial. But the Guinea study showed 100% efficacy. No cases occurred in people who were vaccinated 10 days or more after randomization in the study. It was very well tolerated. And there were only 3 serious adverse events reported out of 6,000 or so subjects. And these were all recovered without sequelae, influenza-like illness, etc. So this, I think the importance here of the precedent set by this in the case of COVID is really important. the COVID vaccines that were developed were based on platforms used to construct Ebola. The chimp-ad vaccine, Adeno 26, chimp-ad now licensed by AstraZeneca for COVID, Ad 26, Johnson & Johnson, and another important feature of that is that a heterologous prime boost strategy was used, really for the first time. So things that happened with Ebola on these viral vectors and on the idea of mix and match or heterologous prime and boost really were important lead-ins to how we managed to move so quickly with COVID vaccine. Now this, next to the last slide, is an eye chart. I'm not going to look at this, but viral vectors are here to stay. They really were evolved, other than the flavivirus vector, which preceded, VSV and MVA and chimp-ad and Ad26. These have started, with Ebola, and now we're instrumental in moving the field forward with COVID. So this is, I think, viral vectors and now, of course, mRNA are the platforms of choice to try to deal with outbreaks of this sort in short periods of time. So what was Phil's role in all of this? He, of course, was in the Department of Defense. He provided oversight and leadership in the Army, and in particular at USAMRIID, where a lot of Ebola work was done and still is. He was at Office of Public Health Preparedness, now ASPR, where he was instrumental in biodefense vaccine development for smallpox and anthrax. Influenced the setting up of BARDA in about 2006, Sabin Foundation, and many more. And during the early stages in 2014 of the VSV vector vaccine development at New Link, he was an advisor. I spoke with him every week, he and Ed Einstein from the Army, and he always listened. He always had fantastic advice, calm view of the big picture, and thanks to Phil for his wisdom and foresight and leadership over the years. Thank you.
Okay, thanks, Tom. Next we have Scott Halstead. So you can just advance from the arrow. To advance.
Magic arrow.
While they set up, there's really a lot of chairs in here for those that are standing up. Please feel free to come forward.
Thank you. Well, that was something. Good morning, everybody. Phil also worked on dengue. In fact, we worked on dengue for 60 plus years. So I'm looking around at this audience, and I can see that a lot of you really would like to learn about dengue. So you're lucky. This is your day. And you're going to be presented by data. So I don't want anybody to cower. Data is just something we have to learn how to live with. But before I do that, I'd like to show a few slides for Phil's kids. This is the house on Soy Rewa D where you lived and where I lived. And this is what it was on a festive occasion when we were bidding farewell to, what did I do here? Oh, sorry. No, I'm just bidding farewell to one of the sergeants. Those of you who've been in the Army know that you wouldn't go anywhere without a sergeant, and you couldn't survive. And we owe everything to them, and that was true in Thailand as well. This is the laboratory, believe it or not, where Phil worked. And this is one of our esteemed Thai colleagues, rather famous, Dr. Ananda Nisalak. And these are some of the Thai colleagues that we all enjoyed working with for many years. In the center here is Dr. Nat Bamara Pawat, who served for almost 20 years as president of the Mahidol University. And on the left is Suchart Jatanasan, who founded epidemiology for Thailand. And on the right is Dr. Charat, who's the dean of the School of Public Health and was our host in Thailand. And here's Phil visiting in 1962, examining a patient with dengue hemorrhagic fever. And here we are a few years later as senior chiefs of the CETO Medical Research Lab, which became AFRIMS, the Armed Forces Research Institute of Medical Sciences. So what about dengue? Well, I'm going to give you a vignette, a scenario of life in dengue and life with Phil and life with me, and what it was like to be in the field. And it's called the Battle of the Presidents. It actually describes two different island outbreaks of dengue. But let's start at the beginning. In late 1950s and early 1960s, the world was confronted by an unexpected occurrence, which was people dying of dengue. Now, there are few people in the audience that lived through World War II, but those of you who read about it will read that there were huge pan-Pacific outbreaks of dengue. Everybody had dengue. The Japanese had dengue. Americans had dengue. We isolated dengue from the Philippines, from Hawaii. But nobody died. So what were kids doing dying of dengue? So that was a research question that led to establishing a lab in Bangkok in the early 1960s. And I arrived first and set up a large prospective cohort, not a cohort, but a prospective hospital-based study. And then Phil came along to follow me, and he set up two really important prospective cohort studies. What we learned from the hospital study was to differentiate the responses to dengue. During World War II, all we had seen was something we call dengue fever. You have aches and pains, and a lot of people, adults who get dengue fever, say exactly this one phrase: "That is the worst disease I've ever had in my life." And there's something about dengue, but we didn't see that in children of Thailand. What we saw was children just suddenly collapsing and cold and clammy and dying. And we divided into definable syndromes. One was called dengue shock syndrome. That was the most severe. And when we characterized the antibody response we could observe in these children that had these different dengue syndromes, those that had the shock syndrome had a classical secondary antibody response. Now, we're going to talk more about that because that was part of what Phil contributed. So here are the basic data. You can see we had large numbers of patients. These are all virologically confirmed. And the association of shock syndrome with secondary dengue infection is highly significant. And I went in 1966 to the Pacific Science Congress, where I presented these data for the first time. Now, unfortunately, I can't blow this picture up, but if I could, you would see everybody who was anybody in arbovirology in the 1960s. Dr. Reeves is here, and Phil, Dr. Hammond, and many others. So, as I said, it was to distinguish the etiology of the disease response. It was critical to come with case definitions, and so we came up with a couple. Dengue hemorrhagic fever was the larger group, and that included patients with fever, positive tourniquet tests as an evidence of hemorrhagic diathesis, low platelets, and evidence of vascular permeability. In our day, we had to infer that from performing a hematocrit, where you take the blood and spin it down, and you see what fraction of all the blood is red blood cells. And when that fraction goes up, it signifies that you're losing fluid out of the circulatory system. So Phil, importantly, brought modern science to actually give a more scientific definition of what a secondary antibody response was. Today, everybody knows that the normal immune response is to go through the production of IgM antibodies, and then they're shut down, and then you substitute with IgG antibodies. We didn't know those things in the 1960s, but Phil did some of the first studies where you take a serum sample and you spin it down in an ultracentrifuge, and the heavier immunoglobulins sink to the bottom. They're in the lower fractions. So here it is. We won't. But if you look at the data on your left, those are the lighter the antibodies, the IgG antibodies, and the data on the right are the heavier. Those are the antibodies that you're finding in columns 1, 2, and 3. So that was an important contribution. Now we had scientific evidence that we could say, yes, this indeed is a secondary antibody response, not just an arbitrary number. So when Phil arrived, they had learned the year before, in 1965, that there was an outbreak on a little island in the Gulf of Siam. And that turned out to be a really fortuitous place to study dengue. And because there had been an outbreak in 1965, with some divine inspiration, maybe Tom Mule can tell us how they acquired divine inspiration, they decided they would invest the time to go down to Koh Samui and establish a prospective cohort study. So they did a year-long study during the year 1966. And now here are some of the attributes of Koh Samui. It's a moderately large populated island, 35,000. The outbreak itself occurred over the months of July to October. But what was amazing about it, frankly, is that there were 3 different dengue viruses isolated on this little island. And look at the infection rate for 1966. 47% of the population was infected. And this is what the outbreak of, what the impact of infecting 35,000 people with dengue 1, 2, and 3. See, you had 1,900 primary infections. I won't go into the gritty detail. How do you know? A lot of work was done here. So 1,900 primary infections, among which there were only 10 who were sick. So you can see that dengue is largely an inapparent disease. There were 2,700 secondary dengue infections, people who were infected in the sequence 1, 2, 1, 3, 1, 4, whatever it is. And among those, there were 85 that were sick. So you can see there's a much higher fraction of secondary infections that are ill. But of those, importantly, 14 had dengue shock syndrome. Now, amazingly enough, nature wasn't finished, and the very next year, Koh Samui had a second consecutive outbreak, or maybe a third. And this time it was dengue 4, another amazing event, frankly. And there were 112 febrile children. You can see that primary infections got 11 children sick. All of the worst case among them was dengue fever. And of the secondary dengue illness, there were 42, and 5 of them were shock syndrome. So we had 2 back-to-back outbreaks where there was very strong evidence that a secondary dengue infection was associated with this severe end of the dengue syndrome. This summarizes those data. So all 4 dengue viruses. I mean, these are the first prospective studies of this brand new syndrome, and they're in the literature. They're published in the American Journal of Tropical Medicine and Hygiene. I'd like to see anybody in the audience that has published a dengue paper that cited these. They're very often not cited, and yet these are, in addition to the hospital data that I did, these are very strong data and very well designed, very well conducted studies. So we knew a lot from these studies. Now, amazingly enough, just a couple of years later, there was a second dengue outbreak on an island, this time in the mid-Pacific. You can see it's east of New Guinea, east of New Zealand, and it's the island of Niue. Now, Niue, as you can see on this slide, it had a population of 4,600, a much smaller population. It had been infected, like almost the entire Pacific population, by dengue 1 in World War II, and the outbreak in 1972 involved 30 to 50% of the population. Now, what was reported from this outbreak was that there were a number of severe cases and fatalities. 12 children had severe febrile disease, and 7 of them died. Many of them had petechial rash and ecchymoses, and among adults there were 11 severe cases and 4 died. Now, nobody was on this island, no scientist was on this island, just a doctor, probably an Australian physician who'd been there for some years. So this was observed by just routinely trained and experienced individuals. And the investigation started 18 months after the outbreak occurred. So there are profound differences in the way these 2 outbreaks were observed. Koh Samui was looked at by experts. They came down from Bangkok. We had the clinicians, we had laboratory people, we had everybody. And Niue, you had no, to begin with, no defined dengue shock occurred, and the outbreak was observed just by a single local physician. Now, amazingly enough, a presidential address before this society with this title appeared the next year. The Emperor's New Clothes Revisited, or Reflections on the Pathogenesis of Dengue Hemorrhagic Fever. And what Dr. Rosen, the evidence that he put forward was that he felt that there was good evidence that dengue shock syndrome had occurred on Niue, and that it was caused by a primary infection. He had shown that dengue 2 had caused an island-wide infection in 1975. He felt that the arguments, some of which you've just seen that support the association between two infections and dengue shock syndrome, those arguments are transparent. And he asked in his speech, did in fact two rogues sell the emperor an invisible garment? So my question to you today is, what's invisible about the Thailand data? We had a large prospective hospital-based laboratory-confirmed study with reported in 12 separate publications. We have the two prospective population-based serological studies that I've just described. So it boils down really to what's what. In retrospect, newly presented children and adults who had a febrile illness. Possibly some of them had dengue. We don't know because we don't have anybody there at the time. I think there was serological evidence of dengue, but do we know that the disease in question was dengue? The physician who saw these patients felt the ones who were severely ill were in shock. The trouble is he didn't define shock. Dengue shock syndrome is really quite a specific syndrome. It's not just, well, golly, the patient is weak and pale and has a weak pulse. The shock syndrome in dengue actually narrows the pulse pressure. Rather than having a normal pulse pressure of 20, you have pulse pressure of 10 or 15. It's very, very specific to dengue. So Rosen said in his presidential address, I'm unaware of any criteria by which dengue infections can be separated into two distinct clinical entities. But if you read what Phil Winter said in the 1968 report of the Thailand outbreak, dengue shock syndrome is the only segment of the dengue clinical spectrum that can be differentiated by clinical examination with reasonable precision. So even today, we're suffering from a case definition problem. We've got a brand new WHO case definition that has sort of eliminated quantitative decision-making, quantitative observations. So, from the perspective of 60 years, thanks, Phil, from the other rogue.
Okay, and for our last talk, Jim LeDuc. So you can just do the arrow.
Okay.
It's already on there.
Well, thank you all very much for coming and for the invitation to participate in this. You're going to hear some stories now that you haven't heard before, I suspect. So one or two. But anyway, I'm going to read this because I want to make sure I touch on everything. And so when Mike first asked me to join this tribute to Phil, he suggested that I focus on Phil's role in nurturing the One Health initiative that's gained widespread acceptance in recent years. And this was an easy fit. Phil had always enjoyed the outdoors. He loved to hunt and fish and sail on the Chesapeake Bay, especially with his good friend Joel Dalrymple. So today I'd like to tell two stories. And these began a half a century ago. These are stories about Phil and Joel and their work together in the legendary Swamp Project, and next, the phenomenal discoveries that Phil facilitated on the hantaviruses. Phil and Joel were like brothers, with Joel on the front lines leading the hands-on work, and Phil, the constant advocate, facilitator, and ultimate cheerleader. As I gathered my thoughts for the talk, it became clear to me that the real story was Phil's great contributions as a leader. Regardless of the topic, Phil could always make you feel that whatever the topic was, it was the most important task ever, that your personal involvement was critical, and that he'd be with you right beside you every step of the way. He was a great motivator who not only knew the science better than anyone, but also, importantly, knew how to get it done. Throughout his career, in uniform and later, he was the driver that made so many great things happen. Phil was one of the first people I met when I reported for duty at RARE in the fall of 1969. Vietnam was raging, soldiers were dying of infections, and Phil was the chief of virus diseases, leading exciting research projects. As a freshly minted Army officer, I was overwhelmed by the exceptionally talented people working there and by the classic RARE building, itself with high ceilings, small offices, and labs with closets that passed as biocontainment facilities. If the walls could talk, oh, the stories that they could tell. The morning chatter around the coffee pot and the lunchtime discussions became the highlight of my time at RARE. Whether it was the latest studies underway in the lab, the play of the Redskins over the past weekend, or what Connie had made for lunch for Phil that day, everything was furiously debated, with Phil leading the charge. His genuine interest in the team at a very personal level was a constant throughout his career, be it as a department chief, hospital commander, commanding general, or a senior government official. Okay, I find the right arrow, yeah. Let me turn to the SWAMP project. In addition to projects on dengue, respiratory diseases, and hepatitis, RARE had a field project underway in the eastern shore of Maryland at the Pocomoke cypress swamp. There had been a series of outbreaks of equine encephalitis, killing ponies on Chincoteague and Aztec Island. Others, like Tom Ewell, may have a better recollection of how the SWAMP project started, but by the time I got there, it was well underway, and Joel was leading the charge. Joel told the story of the early days of the SWAMP project when Phil and Ed Busher were trying to collect blood samples from ducks near Chincoteague to look for evidence of eastern equine encephalitis. But they weren't having much success. After a day of frustration, a local waterman named Tom Reed said in his heavy Chincoteague drawl, "You boys are only catching the dumb ones." Well, to which Phil responded as only he could, "If you're so blankety blankety blank smart, then you do it." Well, the next day, Tom turned up with about 30 ducks, and Phil hired him on the spot. I can't attest to the accuracy of this story, but I can tell you for sure that for the next several years, Tom was an essential part of the SWAMP team. And since everyone else was an MD or a PhD or had other advanced degrees, we all decided that Tom was a PDH, pretty damn handy. This is a photograph of the SWAMP headquarters, if you will. The office there was the old abandoned bus, and that's Ed Sogstead, Tom Reed, Joel, and me. And these are some of the field collections that were done, again, with Bruce Eldridge, Ed Sogstead, in one of the CDC light traps. I'm sorry, you probably can't read that, but the SWAMP study continued for several years and resulted in many key discoveries. In 1969, over 350,000 mosquitoes were collected and tested for viruses and found that only Culicida melanura was infected with eastern or western equine encephalitis. Bird collections and sentinel quail studies demonstrated that peak transmission occurred during July and August, and that white-eyed vireo juvenile birds were a prime host, and that they became infected soon after fledgling. And blood meal analysis of engorged mosquitoes found that Culicida melanura fed almost exclusively on birds. These studies were foundational in our understanding of the local transmission cycle of equine encephalitis viruses on the peninsula. During the equine encephalitis study, several other viruses were isolated. One of the most commonly found was Keystone virus, a little-known member of the California group viruses. We focused on this virus in 1971 and 72, and from 180,000 mosquitoes processed, we isolated 96 viruses, all but 3 being Keystone virus, and all of these were from Aedes atlanticus mosquitoes. It was clear that Keystone virus was closely linked to Aedes atlanticus, and we were able to recover Keystone virus from field-collected larval mosquitoes and from newly emerged wild-caught male and female mosquitoes reared in the laboratory. These convincingly demonstrated transviral transmission of Keystone virus. This was a big deal at the time and even won top honors at the 1974 Army Science Conference. It also complemented work done by other swamp veterans, Doug Watts and Tom Mule, who had made similar observations on La Crosse virus in Wisconsin. The swamp project ended a few years later, and in 1987, Phil hosted a small gathering to honor Tom Reed, and this is a photograph taken at that service. Let me turn now to the hantaviruses. In the late 1970s, it became apparent that the old rare laboratories were inadequate to safely conduct research on the dangerous pathogens of military concern. And much of the work on high-hazard viruses was moved to USAMRIID at Fort Detrick. After a couple of overseas assignments, I was assigned to USAMRIID in 1981, where I joined Joel and others who had already moved there from rare. After more than 2 decades of searching, the cause of Korean hemorrhagic fever had finally been discovered by a Korean scientist, Ho-Wang Lee. Korean hemorrhagic fever had caused serious disease among American and UN forces during the Korean War. And despite the best minds working on it at the time, the cause had remained unknown until Ho-Wang Lee, working under Army sponsorship, finally isolated Hantan virus from field rodents. At this time, very little was known about the virus, and indeed, it wasn't even clear that this, in fact, was the true cause of KHF. Encouraged by Phil, Joel and Ho-Wang began a fruitful collaboration that continued for more than a decade. And this partnership was enhanced by the addition of Connie Smalljohn, a bright and energetic then NRC fellow who joined the team and rapidly demonstrated her remarkable skills as a molecular virologist. This was an exciting time of discovery and adventure that included a team of outstanding scientists, many adventures around the world, and expanding collaborations internationally. Some say this was the golden era of USAMRIID. Despite his military advances, Phil remained dedicated to the hantavirus work, and even as commanding general, he continued to engage in the same fierce discussions of the science with all of us. Spirited dialogue and personal commitment that was evident during the lunches at Rare was still there when we met with the general at command headquarters or over a beer someplace. The decade-long collaborations on the hantaviruses are nothing short of phenomenal. An entirely new taxon of viruses was discovered, soon leading to the development of refined diagnostic capabilities that allowed us to confirm that Hantan virus was, in fact, the cause of Korean hemorrhagic fever. This slide shows the results of IgM and IgG tests on stored sera that had been collected in the 1950s by the Hemorrhagic Fever Commission. This is a slide of Joel and Song Gan, the famous Chinese investigator scientist who had worked on hantaviruses, on epidemic hemorrhagic fever, and along with Joel is Pete Canonico. This was taken about the time we were talking about starting the ribavirin trial in China. One of the most exciting early studies was the ribavirin trial that was conducted in Wuhan, China in the mid-1980s. This was done in the old China relatively soon after the Cultural Revolution, at a time when China was just beginning to engage internationally. Despite many challenges, and I can tell you there were many, the study was a success in demonstrating the efficacy of ribavirin in the treatment of hantavirus infections. And equally important, and perhaps more so, it set in motion scientific partnerships and true friendships that continue to this day. In 1989, we tried to replicate the China study in what was then Yugoslavia. We set up a field lab in the medical school in Sarajevo, but we had to abandon the work due to the Civil War. Nonetheless, we built great partnerships that have remained to this day. And that's, you see Tom Kajak in that photograph here. One of the benefits of the collaboration in Yugoslavia was the discovery of a new hantavirus, Dubrava virus, that caused especially serious and often fatal human infections. Some of the international fellows working at USAMRIID at this time are shown in this slide. And in the center is Tetiana Avcic, who was the lead author in the discovery of Dubrava virus. Another aspect of the hantavirus studies was the discovery of sole virus in domestic rats in the US, and indeed in many places around the world. For a decade, we studied the hantaviruses in Baltimore, and the photo in the lower right shows the three leaders, Jamie Childs, a very young George Korch, and Greg Glass. This is just a snippet of the results that we found, but demonstrated here is the prevalence of sole virus infections among Norway rats captured in Baltimore, with nearly all the rats becoming infected by the time they reached adulthood. We also studied, had exciting collaborations in Sweden on Pumala virus, the cause of a serious disease called Nephropatia epidemica. The hantaviruses became a prime example of a new concept in global health, emerging infectious diseases, and the link to the One Health initiative. A completely new group of viruses was discovered, and the characterization of Hantan virus led to the recognition of many new hantaviruses around the world, including throughout the Americas. Studies to identify drugs and vaccines to protect against hantavirus infections began with these discoveries, and these continue to this day. Importantly, the global engagement that resulted from the hantavirus studies has led to lasting friendships, productive collaborations, and training opportunities for an entire generation of scientists around the world. None of this would have happened were it not for the beautiful collaborations facilitated by Phil and built by Joel and Ho Wang. The very existence of that partnership and all the remarkable studies that ensued was the direct result of Phil's leadership, his vision, and his appreciation for the importance of global engagement. Here's to you, Phil. Thanks for the memories. Thank you.
Well, we did great with time. We have a few minutes if anyone has questions. I think there's a mic in the back. So.
I think we just thank them and send them off.
Okay. Well, if no questions, we really appreciate everyone coming. I think you would agree that Phil Russell is a very special person, not just for all the organizations in which he worked, but for the larger field of global public health. And we appreciate your attendance. And yeah, so thanks for coming.